Okay, our next speaker is Dr. Dhruv Sareen. He has extensive experience both in pluripotent and neural system cell research. He developed and directed the Cedars-Sinai Medical Center IPS core, and recently, just this year, started an independent laboratory. He's published high-profile papers on human iPSC-based disease modeling in spinal muscular atrophy as well as ALS. His group has optimized the generation of non-integrating iPSC lines and have now generated more than 85 IPS lines from patients with many different diseases. Dr. Sareen's research focuses on human IPS technology to understand molecular mechanisms underlying neurodevelopmental degenerative diseases and how obesity is linked to the brain. So please welcome Dr. Dhruv Sareen.
Thank you for the kind introduction. I am not going to focus as much, and I think Lawrence did a wonderful job on explaining technologies for reprogramming IPS cells. Any questions related to that, of how we generate IPS lines, I can probably take at the end, but there won't be as much details on that. I'm going to focus more on the applications of the generation of these IPS cells. All right, so here's a little bit of a background, an outline to the talk. Basically, I'm going to talk about the IPS core, characterization, differentiation, and the new technologies going on. And in the end, talk a little bit about the applications of disease modeling and drug discovery, of how that's actually of interest to my lab as well. So I had a little bit of a provocative title, From Bland to Grand, and the reason it was because the somatic cells in my lab are considered very bland because they can make all the cell types that are of interest, and people have been using fibroblasts to immortalize and study for, in fact, neurodegenerative disorders, whereas, fibroblasts don't make neurons. So the technology that Lawrence talked about, the IPS technology, since the advent in 2006, we've been able to make IPS cells, which I believe are very grand, because of two main properties. They are self-renewing, so you can make billions and billions of them and bank them, and you can specialize them, knowing knowledge of development biology, to make a specialized cell type, for example, a neuron, a muscle, a lung epithelial cell, to study these diseases. So in the Cedars IPS core, early on, we started with lentiviral technology for reprogramming IPS cells, but that quickly dissipated as newer technologies came about. And in the end, we focused on our reliable system was the episomal plasmids in combination with small molecules to enhance the efficiencies of reprogramming. And the reason was because lentiviral technologies, we saw that there are certain genes, for example, NANOG, would not silence completely upon differentiating to certain cell types, for example, neurons. And so this led to variable phenotypes in terms of disease modeling and comparing healthy patient lines versus disease lines. And also our long-term goal was to make clinical grade IPS lines, and that wasn't going to work out with the lentiviral technologies. So our workflow in the core is, we receive, isolate fibroblasts or blood samples. Now we're pretty much focusing on peripheral blood samples and any skin biopsies that we get readily from the informed consents in the clinic. And then reprogram, isolate the clones, expand for about 20 weeks to fully characterize them and create a bank that we can distribute to many researchers. The early banks can be distributed around within 5 weeks post-generation. Characterize lines and then differentiate to confirm that. And then eventually tied to that is a bunch of characterization services that are offered in our core, and along with differentiations, but also we're proud to start a very formal training program in stem cell techniques, because at Cedars before there was no stem cell program, so we've had to train these clinicians and new scientists in doing IPS work and differentiation. So this is Lauren, who's kind of done a lot of the IPS generation. She's the core manager, and she's wonderful. She's here. She's training high school students, so we've got a high school training program as well. So in terms of characterization, this is quickly, we do a very first on step within the first passages of IPS generation, characterization of the pluripotent antigens, so the cell surface and the nuclear antigens shown here. And eventually, within the first 7 to 8 weeks, we move on to a whole genome-based gene chip assay. It's an open access assay that we use to characterize the iPS lines, which is Pluritest. And it's worked wonderfully for us. So far, we've done actually now greater than 150 lines, close to 200 lines. And about the rates stayed about the same, where 85 to 87% of them have passed the Pluritest. And about 15% have come back further evaluated. And that, we think, further evaluate means reason that either it could be a disease that's causing them to have a phenotype in the iPS stage that's causing certain gene expression patterns, or they're partially reprogrammed. And we confirm that by doing further characterization assays. So shown here, just an example of what Pluritest encompasses is this red cloud on the top is high pluripotency score and a low novelty score. So that means they are high in pluripotency genes, and this is based on a mathematical model developed by Mueller and Jeannie Loring at Scripps. So we basically adapt that, and it's published. And so this cloud is pluripotent cell lines, whereas here fibroblasts or neuroprogenitor cells cluster below, and they are very low in terms of this assay. They're not pluripotent. So the other assay which we don't get from Pluritest is how these lines are going to fare when you take them and differentiate them into different cell types. So for example, if we want to make endoderm, mesoderm, or ectoderm, the 3 germ layers of interest, we get that knowledge from the PSC scorecard. And here's an example of 2 clones from 2 lines from a control patient and a spinal muscular atrophy patient that have been made into embryoid bodies, and then we subject them to the Pluritest, the TACMAN PSC scorecard, and then they make all the 3 germ layers, and you get a score for that. So they have the tri-lineage potential. And so this was the early version of this, and then we have this work actually submitted in a manuscript. And the new version of this, and we worked with Deb Tyberg on this to get further handle on the PSC scorecard, and they've done some further innovations in terms of the algorithms behind calculating these scores. And I wanted to show you an example of what happens here, and it's not really evident, but this is the middle 2 columns are iPS lines, so they're blue, and this panel is for looking at the expression of endoderm genes. And so they're blue, but that means that they're low in expression for the endoderm genes, which they should be at the pluripotent state, whereas all the embryoid bodies, day 14 differentiated, are red, so that means they're high expressors for endoderm, so they have a good potential for endoderm. This one line, which was iPS, it says 97I HD, and you can see that it's actually this one right here. It's blue. And we've confirmed this with multiple assays, and this was one of the lines that actually scored further evaluated in Pluritest and also came out as not being able, so it had a bilineage potential and could not make endoderm. And so we were able to quantify that either it's something to do with a disease phenotype or it's a partially reprogrammed line. But all our other characteristics say that by flow cytometry and other gene expression studies that it expresses all the correct gene expression profiles for it to be pluripotent. So it's something probably inherent to the disease phenotype. So in the core, then after we make these cells, then we pass it on to researchers or in my lab. And so I'm specifically interested in studying patient neural stem cells to look at the early neurodevelopment in a very disease context of certain mutations, for example, NF1 and RAS. And so shown here is an example of the red is a mitotracker, and the green is a genetically zinc finger targeted iPS cell line that expresses a nuclear GFP under the beta-actin promoter, and that was developed in the core. So it's ubiquitous. Since beta-actin is a ubiquitous promoter, nuclear GFP is always expressed, and so we can always track these lines. So then these neural stem cells are looked for their mitochondrial trafficking properties. So this is a disease I'm interested in. There's mitochondrial deficits. And once that happens, we take them into certain type of neurons. So in this case, these are motor neurons that are made using caudalizing and ventralizing these neural stem cells. And then eventually we stain and characterize them for the correct markers of motor neurons. So this is SMI32, or neurofilament heavy, that's a motor neuron marker. Eventually, we pass it on to other collaborators, and this is work by Rob Barrett and Steph Targan in the Institute, where they take these iPS cell lines and make them into these 3D or intestinal organoid structures, and they can confirm that they make endoderm subtypes that are representative of all the lineages of gut epithelial cells. And then they want to go on, take this on and study inflammatory bowel disease and use these intestinal epithelium and organoids to study influences of how IBD is affected in the patients. And then this is, again, the GFP line that I was talking about. And then we can go on and, using a very simple 14-day protocol, make them into these cardiac mesoderm organoids, or cardiac EBs, that are plated down. And you can see them beating. So eventually the goal is to give it to our collaborators in the Heart Institute and ask them to screen for certain cardiotoxic drugs to see if there's certain patterns of arrhythmia in these cardiac organoids. The other thing that's going on in the core is we're really improving the technologies, and we're using high content imaging to screen for multiple modalities of reprogramming and speed up our reprogramming process. And what we've shown is that we've got now a defined media that in 14 days we can get iPS colonies in, and we just need 6 wells of a 96 well plate to get enough colonies. And in each well there's a single colony. So our eventual goal is to start freezing these 96 well plates down and pick the best clones and isolate and expand them early on. And whatever's left over, we can actually bank them in case we need to ever go back at a later stage. And this is showing in the 96 well plate at day 30 that we get these beautiful NANOG and SSCA4 positive colonies using our method. And showing an example of this on a little bit of a larger scale. So we had one fibroblast line that was really difficult to reprogram with the episomal plasmid method, just using episomal plasmids, and alkaline phosphatase is shown here as a marker of pluripotency. And when we used our traditional protocol using the plasmid and cocktails, we were able to get a few colonies, about 6 or 7 colonies per input of 400,000 cells. When we use this defined media with plasmids, first type of defined media that we tried, we got some colonies, but the expression of alkaline phosphatase was low. But you can see that once we switched to this defined media and removed some signals, specific signals for pathways, that we were able to take this and reprogram them into iPS cells really efficiently, from just episomal plasmids to plasmids in the defined media. So media could just get them to over the hump for making iPS cells efficiently. So we're taking this to the fact that now we're efficiently reprogramming peripheral blood mononuclear cells, both alpha T cells, alpha beta T cells, and the non-T cells. And the non-T cells are most likely monocytes. I should say the efficiency of reprogramming non-T cells is tenfold lower than T cells. But we can do this from a simple blood draw of 8 mL, 4 to 8 mL blood draw. We can reprogram iPS cells using the plasmid-based method really efficiently. And I should mention that there's a lot of lymphoblastoid cell line repositories with clinical genotype and phenotype that are severely being underutilized for reprogramming iPS cells, and we have clinical data on them. So, if there's interest in correlating clinical data, genotype, to iPS cells and the cell types that we can make from them, then we've used this technique now to make lymphoblastoid cell lines really reliably into iPS cells. And so what we've shown is using this modified protocol, this cocktail of plasmids, and this work's submitted it's under review right now. So we can make, within 25 days, we get iPS cells really efficiently. And so far with this method, we've had about 100% success. So all the 17 lines that we've tried, we've been able to make into iPS cells, albeit at different reprogramming efficiencies. And so we've been really fortunate to do this with our Cedars banks of IBD lines, SMA lines, and BRCA1 lines. And so what we went on to show that these lines then eventually make neurons, in this case, and the intestinal epithelial cells that I talked about earlier, pretty similar to any fibroblast-derived iPS cell line. So there was any questions regarding if there's residual epigenetic memory and then maybe blood cells may not make certain cell types. We answered that in terms of making at least a couple of different cell types and showing that they're equally efficient in making them. So just to conclude this part of the talk, so we made about greater than 85 iPS cell lines, and they're available and listed on our repository from us, and we just recover our costs. And they're funded by different grants or institutional mechanisms. And so they're available for after certain days. We're ready to provide these cell lines to investigators. And yes, we use different tissue sources, but primarily we're focusing on fibroblast, lymphoblastoid cell lines, and peripheral blood. So in terms of the applications that I'm using to in my lab and within the institute to use iPS cell lines. So since we've started working on iPS cells in the last 2 to 3 years, we've had a number of publications in here. This is in SMA showing that the iPS cells can remodel and faithfully remodel SMA phenotypes, Huntington disease, amyotrophic lateral sclerosis, Lou Gehrig's, and collaborators has done this with skeletal dysplasia. And so we've also taken this down to develop more techniques in cell therapy where we've shown that we can make neural progenitor cells that survive and integrate into the spinal cord of rats, and then they survive and migrate as efficiently as the fetal-derived progenitor cells that have been extensively investigated. So, and then there's other methods for using these iPS cells. But specifically, I'm interested in motor neuron disorders. And just to give a little bit background, there's motor neurons are classified into upper motor neurons that connect from the brain to the spinal cord, and then from the spinal cord to the effector muscle. And in 2 different diseases, in SMA, which is a pediatric neuromuscular disorder, the lower motor neurons are primarily affected, whereas in ALS or Lou Gehrig's disease, both lower and upper motor neurons are affected. So, and they both go through this degeneration phase. The mechanisms are currently, there's many mechanisms purported, but there's a non-cell autonomous mechanism where there's contribution of glial cells and microglia also reported to be known. So I'm interested in screening for high-content screening to find molecules or small molecules that would revert the SMA phenotype. So in the normal, this is a mouse model showing the spinal cord of a normal mouse, and you can see these are the motor neurons stained in the bright blue with Nissl. This is a normal, whereas in a SMA mouse model, you can see that the motor neurons severely degenerate. So the idea is to screen for a second modifier gene that is intact in these patients, and that can produce almost full-length functional protein. So the idea is to change this modifier using small molecules so that it produces more of that functional protein, which is SMN. And SMN in the nucleus forms these gems. So we try to make an assay where we identify these gems only in motor neurons and screen for drugs that identify these gems in these motor neurons. So what's thought that in these gems there are functional units and they're involved in splicing machinery. So the challenge was to take these beautiful motor neurons that we can grow in low throughput in a dish and taking them into high content and making billions of them, or almost 500 million of them, to screen annotated chemical libraries. So we devised a protocol to make these motor neurons reliably and expand them at different stages and freeze them down. So first step was to take iPS cells, make neural rosettes, which is akin to neurodevelopment and where they form neural tubes, isolate those neural rosettes, and then cautoventralize them and expand them in mitogens. And then we use this chopping method where we have an automated chopper to chop these spheres and expand them. So here's shown characterization of sections of these spheres where they have all the markers for motor neuron precursors, shown with islet 1, NKX, LHX, SMI32, and they're proliferative. So at this stage, they can proliferate, and so we've reliably shown it with 10 or more cell lines that we can make this into a reliable method for freezing them down. And then eventually we can plate them down and dissociate and plate them down and seed them in these 96-well plates. But even then, that's a very long protocol. That's still another 30 days in a 384 96-well plate. So we're now working to optimize that second stage where we can freeze them down and cryopreserve them in billions of quantities at about 15 days post-plating. So here's shown that these are from control, SMN and ALS line. We can make these cells. This is TUJ1, which is a neuronal marker, SMI32, HB9, and CHAT are motor neuron markers. So this is images from 384 well plate format. So the second goal is that in the motor neurons, we want to show that they still have this phenotype where the SMN protein's downregulated and the SMN transcript is still downregulated. So the goal is to get this SMN protein back to the normal levels, and we want to use that SMN assay, GEM assay, as a surrogate for taking these further and finding novel drug candidates that can be used in motor neurons. So we've developed, in collaboration with molecular devices, to develop a high-content imaging assay. So here is showing motor neurons in a culture, and then SMN is staining for that marker GEM in that motor neuron. And we've developed an algorithm to identify motor neurons and then segregate them specifically in the nuclei of motor neurons and then quantify those motor neurons. And so currently, as we're expanding the cell lines, in the next month, we're gearing up to do some 300 compound screen, not 300, sorry, I meant about 3,000 compound screens from an annotated library to identify candidates. So that would be the primary screen, and then a secondary screen would be to reverse the phenotype that's actually seen in these SMA motor neurons. So here's two assays that we're going to use for the secondary screens. One is that the SMA cells have a delay in the neurite outgrowth, so they don't show same level of neurite outgrowth as compared to the control cells. So we'd find we're going to be looking for compounds that eventually from the primary screen would reverse this phenotype to get full neurite outgrowth potential. And in response to stress, the SMA cells are much more sensitive to withdrawal of this neurotrophin, BDNF, compared to the control cell lines. And so we're going to look for this reversal of phenotype as shown here. You'll see a lot of annexin 5 staining in SMA cultures when you withdraw BDNF compared to the control cells. So we're going to try to look for withdrawal of this phenotype. So in conclusion, I believe that iPS cells are a very versatile tool for recapitulating human diseases. And we've shown an application of that with high-content screening to make motor neurons reliably from these iPS cells in large quantities, and we've confirmed their function. I didn't show data on that. We've confirmed function by EFIS, calcium imaging, and multi-electrode arrays. And now we're gearing up for screens for about 1,400 to 3,000 compounds. And so in the end, I would like to thank all the people who do all the hard work in the core. So this is our team. They're the minions dressed on Halloween here. But this is the IPS core facility, all the funding that we have, and all our collaborators in the core and internationally. So majority of the work was done by Lauren, Burhan, Lindsay, and Ice in the core. Thank you. And I'll take any questions.
Good. That was a nice presentation. Thanks for sharing that with us. I took some notes. I was kind of interested on the characterization protocol you guys use for iPS cells. And I see you use hematopoietic stem cell scorecards for TACMAN and use that taking material from embryonic bodies. And how reproducible would be that when you see that an embryonic body can actually get a high score for endoderm or mesoderm or ectoderm? And if you do that protocol over and over again, you would get the same results, and is this kind of reproducible?
Yeah, so that's a good question. So we have seen, the kit's relatively new, but we have done a couple of cell lines the same times, and the one that I showed that wasn't able to make endoderm, that has been reproducible. So that cell line would definitely not make endoderm. Now, I'm not very confident as to saying, if you make embryonic bodies from the same cell line at passage 10 and 20, and you get a score of, say, 25, and if you get 20 the next time, I'm not confident in saying that, and maybe I think life can help with that a little bit. But we've seen that the same cell line, if it's deficient in making a certain cell type, it's always deficient in making that cell type.
And how probable would it be that a certain type of score, being that the Stackman assay or any other ones you guys are used to doing in your facility, to replace teratoma assay? Because for sure, to characterization.
Good question. No, so we haven't done teratomas for 2 years.
Okay.
And like I said, all the manuscripts I showed were without teratomas.
All right.
So, and reviewers had problems. We argued. We won.
Okay. That's cool. Thank you.
Yeah.
We have several questions coming in from online. One was several around reprogramming. Are there specific disease samples or donor profiles that you found to be particularly resistant to reprogramming that you can categorize, or has it been random in your understanding?
It's been resistant to reprogramming, probably not. So yes, okay, I should go back. So if there's starting fibroblast samples is really senescent. So for example, if they're not mitotically active and they're not dividing, then they're really resistant to reprogramming, and we've had a hard time reprogramming those. But there are certain disease contexts where we've been able to reprogram efficiently, but we have a hard time maintaining them in an undifferentiated state, and that's kind of disease dependent. So each cell line has a tendency to spontaneously differentiate it a bit, and so we have a wide spectrum of really good cell lines and really tough to maintain cell lines, and that's disease dependent.
Okay. And one more here. Have you assessed, or do you plan to assess, whether your motor neurons are capable of forming neuromuscular junctions?
So that's a good question. So we're actively doing that. And preliminary data is that assay is a little bit difficult because to make iPS-derived skeletal muscles, still now, the protocols haven't been well established. So we've used co-cultured motor neurons with muscle cells derived from from a different source. And then the culture media that they grow in and the co-cultures are very difficult. So they're not quite compatible. So the muscle cells don't like the motor neuron media, and the motor neurons don't like the muscle media. So we're trying to segregate them in devices and do muscular assays, and that's under process. So preliminary results are very positive, but I don't want to get too optimistic about that.
Okay. Well, with that, we will close the session. Thank you all for attending. Thank you, Dr. Sareen, for your presentation.